Good afternoon. Has anyone taken Professor Moshewitz's immunology course? Some people. How about you people listening? Any of you? Nobody, I guess. Well, today we're going to talk about host defense. One lecture for host defense. So obviously, this for many of you will be a repeat, although maybe not with the viral emphasis that I'm going to give it to you today. And for the others, there's a lot of information in the textbook that can fill in the gaps. So we view the host defense against infections in sort of three stages, if you will. The first being the physical and chemical defenses that are nonspecific, your skin, surface coatings of mucous membranes, low pH, etc. Then what we call the frontline defense, the intrinsic defense systems, which you'll hear more about from Professor Silverstein, the detection by the innate immune system, production of cytokines, and so forth, and then finally the attack and cleanup, the adaptive immune defenses led by antibodies and T cells of various sorts. Another way to look at it is shown here. You have a pathogen impinging on us. Not shown here are the intrinsic defenses, the non-specific ones. If the pathogen gets by that, it's recognized by pattern recognition receptors. If there is no recognition as a pathogen, that's the end. But if it is recognized as being foreign, and we'll talk a bit about how that happens, this activates a number of arms. The cytokines are produced, dendritic cells are recruited in part via the cytokines, and this comprises the innate response to infection. If the pathogen would happen to bypass the innate response, then the adaptive response is engaged, and this results in the end in the production of very specific B and T cells, which hopefully will eliminate the pathogen, and eventually the establishment of memory of that pathogen, so that if it's encountered again, you have a very rapid response. I view the immune system in this way: three walls of higher height. The first is the intrinsic defenses. If a virus gets over that, it encounters the innate immunity wall, and if it gets over that, then we have acquired immunity. And then if it gets over that, then unfortunately the host is in big trouble. So let's start with the innate immune responses. We're going to bypass a lot of the intrinsic responses, as I said. Dr. Silverstein will talk about those. Innate immune responses are always active in the host without any exposure to a virus, and they are typically activated very quickly after infection, and it's the only immune defense available for the first few days after an infection. So until your adaptive defenses kick in, you're at the mercy of your innate system, and so it's very, very important. The innate response comprises cytokines, which we'll talk about, sentinel cells, including dendritic cells and macrophages, complement, and NK, or natural killer cells. And the activation of the innate response relies on the recognition of a foreign pathogen by various patterns. And these are molecular motifs that are found only in microbes. And as I said earlier, the innate response is able to communicate with the adaptive response, and it will inform it when replication has exceeded a certain threshold and the innate response can't handle it any longer. An important component of the innate response is recognizing that a microbe is foreign and not self. And this is something that has only developed in the past 30 years or so. This story begins in 1980 when two German scientists, they're working on Drosophila, not viruses. They were trying to identify genes involved in pattern development in flies. And the story goes that Christiane Nusslein-Volhard looked at a fly one day, and it was really weirdly malformed. So she said, "Das war ja toll," which means, wow, that's pretty cool or far out. And so she ended up calling them the toll genes that controlled this particular phenotype. And this was a very important finding, and she got the Nobel Prize for that in 1995. So we call them the toll receptors now. In 1996, this gene, the toll gene, was then found to have a role in immunity of the fly to fungal infections. So for all this period from 1980 to 96, we knew it had a role in development of the fly, but only in 96 shown to have a role in innate immunity. So flies just have an innate immune system, so this is an important component of it. And in 1997, toll-like receptors were identified in mammals, and that's what we're going to talk about today, toll-like receptors. These are type 1 transmembrane proteins. They are pattern recognition receptors. They recognize a pathogen-associated molecular pattern, also called a PAMP, and they are conserved from plants, insects, all the way to humans. Highly conserved proteins. 14 members that we know about. The extracellular domains consist of what we call leucine-rich repeats. They are just repeats of a leucine-rich domain. And the cytoplasmic domains are related to similar domains in the interleukin-1 receptor family, so they're called Toll/IL-1 receptor domains. These cytoplasmic domains or TIR domains. So these are the toll-like receptors known so far and the patterns that they recognize. So these are patterns that are specific for microbes, not us. So these TLRs recognize these individual patterns as foreign. And the ones we will consider mainly for viruses are TLR2, viral glycoproteins, double-stranded RNA, TLR3, viral glycoproteins, also TLR4, and then TLR7 and 8 recognize single-stranded RNA. The other TLRs recognize components of bacteria and fungi. So again, these are components that are not within us, but are recognized as foreign. So here are some diagrams of the TLRs. Here's the original protein encoded by the toll gene of Drosophila called Spetzel. And you can see it is a transmembrane protein. It has a cytoplasmic TIR domain right here, and then the extracellular domain composed of many of these leucine-rich repeats. And here are all the TLRs in other species from TLR1 to 10, and some of the components that they recognize here. So again, composed of different arrangements of leucine-rich repeats. The crystal structure of the extracellular domain of toll-like receptor 3 has been solved. You can see it here. Each of these lines essentially is a leucine-rich repeat, and you can see it forms a curved molecule. So this recognizes these foreign antigens and RNAs. Now the toll-like receptors can be located in different places on cells. Some of them are in the plasma membranes and recognize viral components in the extracellular fluids, but many of them are on the inner endosome membrane. So here is a cell. Here's the cytoplasm in the nucleus, and this would be an endosome. And you can see the TLR3, 7, 8, and 9, all of these recognizing RNA, are on the interior of the endosome, so that when viruses are endocytosed and they deposit their nucleic acid into the endosome, or when dying cell pieces are taken up by the cell, the nucleic acid ends up in the endosome, it can be sensed by the toll-like receptors. And that sensing then initiates a signal transduction pathway involving phosphorylation of various transcription proteins, IRF3 and FKB, their entry into the nucleus, and then the production of cytokines, including interferons. So some of these cytokines are produced. So that's the end result of when a TLR senses something being foreign. It initiates a signaling cascade, and you have the production of cytokines, which are a key component of the innate response. Now, how this works is not entirely understood yet, how, say, a ligand activates a toll-like receptor. The current model for RNA, at least, which is shown here, is that the extracellular domain of TLR, this is TLR3, binds RNA, and that's the RNA is shown here as a gray coil. And then the binding of RNA leads to dimerization of the TLR, and that is important for signaling. So the dimerization of monomers of TLR3 here initiate the signaling, which will then produce the cytokines in the cell. There is, in addition to these TLRs, which are membrane-bound sensors, there are two cytoplasmic proteins that also sense viral RNAs. One of them is shown here. It's called RIG-I. And it is, as I said, in the cytoplasm, and it senses the presence of viral RNAs in the cytoplasm. Not every virus puts its RNA or DNA into the endosome. And to cover that, we have these cytoplasmic helicases. These are called Rig-I-like helicases. One is Rig-I and the other is MDA5. So these would recognize viral RNA as specifically being foreign. And that also initiates a signaling cascade which leads to the production of inflammatory cytokines. So we have two systems, a membrane-bound system and a cytoplasmic system for sensing molecules as foreign. This is a model for how these cytoplasmic sensors would detect RNA as foreign. These, both Rig-I and MDA5, they both are very similar in structure. And here's a model on the left for what they look like. They have an N-terminal card domain. They have a helicase domain in the central portion, at the end a C-terminal domain. And they are present in the cell in an inactive form. They cannot signal. They cannot turn on cytokine synthesis in this form. When they recognize viral RNA, the viral RNA binds the C-terminal domain, unfolds the protein, activates it so that it can now participate in signaling. And the card domain is essential for that. The card domain interacts with downstream proteins, and that is exposed when these proteins bind RNA. The way that the RNA is recognized as foreign is that these proteins recognize either double-stranded RNA, which is typically not present in an uninfected cell. So many viruses make double-stranded RNA during transcription or replication. So if that's sensed, these proteins assume it's foreign, and then they initiate the induction of cytokines. Or RNA, single-stranded RNA with 5' phosphates. This is, again, not something that is found in cells. Cellular messages have caps, and other RNAs, ribosomal RNAs, are typically confined to the nucleus. So the only RNA with 5' phosphate in the cytoplasm is going to be a viral RNA. So again, that's why these sensors classify it as foreign. So here's another view of this pathway to give you a little more detail. Here is Rig-I and MDA5 recognizing various kinds of RNAs. They could be negative or positive stranded. It doesn't matter as long as they have phosphates, or they can be double-stranded. When these sensors are activated by binding to RNA, that card domain then interacts with a mitochondrial protein called MAVs. And then that initiates the signaling through a variety of pathways, leading to activation of transcription proteins, IRF3, IRF7, NF-kappa B. These go in the nucleus, and here we're showing the production of interferon by assembly of a variety of these proteins on the promoter. So the point here is that they sense the RNA in the cytoplasm, and then the end result is the production of cytokines. And one of those cytokines is interferon, or interferons, which is a family of proteins. These were first discovered in 1957 by Isaacs and Lindemann, who found that if you expose chicken cells to an activated influenza virus, those cells would produce something in the medium, which if you added to another cell would protect them from influenza virus infection. So they said it interfered with infection, so they called it interferon. There are a couple of different types, as we'll show you in a moment. Interferon alpha, beta are the type 1 interferons. These appear first after infection, followed by interferon gamma. Virus-infected cells typically induce the synthesis of interferon alpha, beta. There are, as part of the innate system, what we call sentinel cells, macrophages, and dendritic cells. These produce alpha beta interferon in response to products released from infected cells. So as we'll see, dendritic cells will scavenge the pieces of dead cells that result from a viral infection, like nucleic acid. They will sense that nucleic acid, and then they will produce interferon alpha beta. So you don't have to infect a cell to produce interferons. The sentinel cells can make it after sensing the products of a dead cell. So here are 4 different types of interferon that we are aware of: interferon alpha and beta. These are the type 1 interferons. Alpha is made in most cells, and it's induced by virus infection. Interferon beta as well, most cells induced by virus infection. Interferon gamma is mainly made by T cells and natural killer cells, and it is induced by T cell receptor activation, which happens when a T cell engages a ligand, and these two cytokines in particular. We've learned most recently of a type 3 interferon called interferon lambda, mainly produced by epithelial cells. So the respiratory tract and alimentary tract epithelium, remember, is a good place for viruses to enter, and apparently those cells have their own special kind of interferon that protects them. So we're not going to talk very much about this, but this is a brand new and very intense area of research. The production of type 1 interferons, that is alpha beta, is very quick after infection, within hours. So you have a burst of interferon production and then it declines by 10 hours. And alpha, so there's one gene for interferon beta, but for alpha there are many. And these seem to be differentially expressed. In other words, different sets of alpha genes are expressed depending on the virus. So the interferon beta, as I said, is stimulated only for a short period of time. So by 10 hours, the synthesis of protein declines. Now, these proteins work on cells that have receptors for them. So interferon alpha will only affect a cell that has a receptor for interferon alpha. In fact, alpha and beta share the same receptor, the type 1 interferon receptor. What these proteins do is to induce the synthesis of still more proteins. And by doing so, they make what we call an antiviral state. So we say the interferon is produced in response to virus infection, and the interferon makes an antiviral state. And that happens when interferon binds to receptors on cells, and that induces the production of over 1,000 cell proteins, which are encoded by genes called interferon-stimulated genes, or ISGs. So the ISGs are the genes whose transcription is stimulated by interferon. They, in turn, make mRNAs that are translated to make all these proteins. So all of these proteins then are involved in establishing an antiviral state. Most of these, we have no idea how they work, these ISGs. It's only in the last few years or so that we've known that there's over 1,000. It started years ago with a few, and then 10, 20, 50, 100, and then with microarray technology, it went over 1,000. So most of them are unknown, and this is a really active area of research. And probably it's true that mixtures of different ISGs are made in response to different viruses, and, or I should say, are effective for inhibiting different virus infections. So what happens is when interferon is produced by an infected cell, say, or even by an uninfected dendritic cell, it diffuses out of the cell, it's secreted from the cell, and then binds to interferon receptors. These can be on the same cell or on another cell. Doesn't really matter. So here we have the type 1 interferon receptor for interferon alpha beta is in the middle, and here's interferon gamma receptor. The binding of interferon to these receptors leads to association with protein kinases, the JAK and TYK protein kinases. Those phosphorylate the STAT proteins, which then dimerize and go into the nucleus and activate the transcription of the ISGs. All right, so another signaling cascade from the binding of interferon to its receptor to the production of transcripts encoding all of the 1,000 or more ISGs. So that happens with both interferon alpha, beta, and gamma, as shown here. So let's talk about a few of these interferon-induced proteins. One of them is PKR, which we've encountered already. We talked about this during translation. This is the double-stranded RNA activated protein kinase. So it's induced by interferon. Its mRNA is induced by interferon. And then the PKR itself, of course, has to be activated by double-stranded RNA. So it then is activated, it phosphorylates the IF2 alpha, inhibits translation. So this is a major ISG because, remember, every virus has evolved a way to get around the activity of PKR. It can inhibit very many viruses, and also the phosphorylated form triggers programmed cell death, which is another intrinsic response to infection. Kill the cell and try and limit virus replication. Another interferon-induced protein is RNase L. There are two of them here, RNase L and 2', 5' oligo A synthetase. So these are two separate proteins encoded by two genes which work together. So what happens here is, again, a virus infects a cell, interferons made, and this is one of the these are two of the mRNAs that are turned on. They're made into proteins. 2' 5' OAS is inactive as it's produced. It's activated by viral RNA, double-stranded viral RNA. Once it's activated, it uses ATP as a substrate and synthesizes 2' 5' oligo A. So it polymerizes A in a very specific way. That product is then required to activate RNase L. So this is an enzyme that degrades viral RNA. It's inactive as produced, is induced by interferon, but in the presence of 2' 5' oligo A, it can become activated, and then it degrades mRNA. Okay? So it's a two-component system, which I suppose ensures that it's only turned on in a virus-infected cell, and the end result is degradation of mRNA. MX proteins are also interferon-induced. These were first discovered in mice, and it was shown to localize in the nucleus and block the cap snatching by the virus. Remember, cap snatching is needed to prime synthesis of viral mRNAs. So this protein blocks that by inhibiting the PB2 protein of the virus. However, mice don't have influenza viruses, and in nature, we can infect them in the lab, which is how this work was done, but in nature there are no flu viruses of mice, and a good amount of the mouse population lacks the gene encoding this protein. So at least in mice, it doesn't have much to do with influenza antiviral activity. But there are human homologs, MXA and MXB, and the MXA protein blocks the replication of a couple of different viruses, as you see here. So a nice example of how studying antiviral effects in mice led to identification of human genes with the same activity. Nitric oxide synthase is another interferon-induced gene product. It's induced by interferon gamma, not alpha beta. And a nitric oxide synthase, shown here in the blue box, takes arginine and makes nitric oxide from it. And nitric oxide has antiviral effects either on its own, or it can be further converted to peroxynitrite, which is a highly reactive compound. Either one, nitric oxide or peroxynitrite, both react with proteins and destroy their activity. So here you can see nitric oxide is reacting with this cloud-like protein here, and it's rendering it inactive. So it's nitrated, basically, and this protein is inactive. So in an infection, what happens is this enzyme is induced, nitric oxide is produced, and it's typically produced locally, right near the infected area. So here we have a epithelial cell monolayer infected by a virus. Here, little virus particles around here. The infection has stimulated the influx of macrophages or NK cells into this area, coming from the circulation to the infected cell. These now produce nitric oxide, and they kill these cells, basically. So they're destroying their own cells, but in the process, they're preventing virus production. So the loss of some cells is considered, evolutionarily at least, not a bad exchange for stopping the virus infection. Okay, so this results in local eradication of infected cells. These do cause pathology, as you would expect. In fact, in mice, you can make mice that don't have the gene for nitric oxide synthase, and for some virus infections, there's less damage, tissue damage in those animals than if they had the gene. So it's responsible for some of the pathology of virus infection, but it's evolutionarily conserved because it must have a selective advantage. Promyelocytic proteins, or PMLs, are also interferon-induced genes. I think you'll hear more about these from Dr. Silverstein. These are present in the nucleoplasm and nuclear bodies. They're called by a variety of names: PML bodies, ND10s, pods. What they do is they bind foreign DNA, and they exert an antiviral effect by inhibiting transcription and the remodeling of nucleosomes. And many virus infections disrupt the formation of PML bodies because they would have an antiviral effect by inhibiting DNA replication. So they are a common target of viral antagonists. A number of components of the ubiquitin-proteasome axis are ISGs. For example, ISG15 is a protein much like ubiquitin that is conjugated to cell proteins that are involved in a variety of activities that viruses need, and this leads to their degradation. So and there are also ubiquitin ligases that are a part of that are also ISGs, and these are of course enzymes that attach ubiquitin to proteins. This targets them for degradation. So the idea being you target viral proteins for degradation, and that helps with the antiviral response. And you can show that these have some consequence because you can add inhibitors of the proteasome. So when you ubiquitinate a protein, it targets it to the proteasome, which is a massive center in the cell for degrading proteins. And if you add inhibitors of the proteasome, you can show that you block the antiviral action of interferon, in this case hepatitis B virus. So these have antiviral effects, and that's why we think they are ISGs. Now here's a very new ISG recently understood. It's called tetherin, or CD137. This is a very interesting ISG. It is actually a transmembrane protein. Here's the configuration on the upper left. Here's the cell membrane. And this tetherin, the N-terminus is in the cytoplasm. And then it crosses the membrane and has an extracellular domain. And then it's anchored, its C-terminus is anchored in the plasma membrane as well. This protein has the property of being able to prevent the budding of many envelope viruses from cells. I'm going to shut the light off a moment so you can see this. Here's an electron micrograph of cells expressing tetherin. The tetherin has been stained so that it appears here as black dots. So this is all tetherin. And you can see these virions are stuck at the surface. And here's on the right is a schematic of how this works. The tetherins are the green molecules. So they're hooking the viruses to the surface and to each other, so they can't move off of the cell and they can't spread. So this has a distinct antiviral activity and inhibits retroviruses and a couple of other envelope viruses. Now, as you might guess, viruses have evolved ways to antagonize tetherin as well. Okay, some other ISGs. ISG56. This is a protein that binds a subunit of EIF3. Remember, EIF3 is a protein involved in translation. It binds EIF4G and it bridges EIF4G and the 40S subunit, so it helps bring in the 40S subunit to mRNA. So the effect of this binding to EIF3 is that it blocks protein synthesis. This is not virus specific, but again, many of these ISGs are not specific for virus functions. They shut down cellular functions, and that includes the virus because, as you know, viruses are parasites of cells, and that stops the infection. MicroRNAs are actually induced by interferon. Treatment of hepatoma cells with interferon beta induces the production of numerous microRNAs, 8 of which inhibit the replication of hepatitis C virus. So it's not a protein, but it's an interferon-stimulated gene, microRNAs. Interestingly, interferon beta treatment of cells reduces the expression of miR-122. And I believe we talked about this a few lectures ago. miR-122 is a cellular microRNA that is actually required for the replication of hepatitis C virus, and it's a liver-specific microRNA. So interferon reduces the expression of this. It's not an ISG in the sense, but it's still an effective interferon that has an antiviral effect. Very interesting. Okay, let's move on now to the sentinel cells, another part of the innate response. These are dendritic cells and macrophages. These exist in peripheral compartments, your skin, mucosal surfaces, first described as Langerhans cells in the late 19th century, and then in 1973, Ralph Steinman and Zanvil Cohn here in New York City called them dendritic cells. And these exist as immature cells initially. They're found in the periphery, body cavities, under mucosal surfaces. So remember the nice mucosa that are beautiful sites of virus entry. These sentinels are below them. They're always patrolling, making sure that nothing bad is happening. They have a whole variety of receptors, toll-like receptors. They have the Rigi-like helicases in their cytoplasm. They have cytokine receptors, and we'll see what those are for in a moment. So they're present in the periphery and body cavities as immature DCs. When they sense something that shouldn't be there, they get activated, they mature, and they go to your lymph node, and there they talk to lymphocytes. They say, look what I found. Look at this antigen. What do you think about this? And then the T cells, the other lymphocytes take over from there. So it's a way of linking the innate and adaptive responses. If the innate can't handle the infection, you've got the adaptive response ready to kick in. So the dendritic cells are important links between the two. They can also make a lot of interferon. So they can sense, they can be underneath that mucosal layer. They can sense virus-infected cells by the products of dying cells. They can sense viral RNA, for example. And they can produce a lot of interferon to try and protect that mucosa before they run off into the lymph node to inform the lymphocyte soldiers. So on the left here is a diagram of an immature dendritic cell. Nothing particularly unusual about it, except it has a lot of interesting receptors. It has toll-like receptors on the surfaces and also in the endosomes. It has receptors for various cytokines that will help mature it. And in its endosome, it has MHC class II receptors. So when this dendritic cell encounters, say, the products of a killed cell at a mucosal layer, it will endocytose. It's very good at taking up endocytosing material. It will detect it as foreign by these TLRs and RLRs, and then it becomes matured. And it becomes this very nice-looking spidery type of cell shown on the right. It's got a lot more surface area, so it can display a lot more receptors for maturation. All these class II molecules are now on the cell surface, and they are presenting these peptides that they found at the mucosal layer to T cells in the lymph node. So they move to the lymph node from the periphery. They present the antigen to the T cell, and they also produce cytokines that will help the T cell mature. And if the T cell recognizes this as a foreign peptide, then boom, it starts becoming activated and then becomes an activated T cell for going and getting rid of the virus infection. And we'll talk about that in a bit. So these guys are sentinels. They hang out in different areas, and when there's something produced that it shouldn't be, they take it up and present it to T cells in the lymph node. Now, some viruses, just an aside here, some viruses can do things to dendritic cells. So you don't have to infect a dendritic cell to activate it. In fact, the dendritic cell just picks up pieces of viral destruction in the periphery and gets activated. But some viruses actually infect them, and it's very smart to do this. For example, HIV-1 binds a protein on the surface of DCs called DC sign. All right, it's just a protein that's there for other reasons, but HIV has evolved to attach to it. It doesn't go into the DC, it just stays stuck on it. The dendritic cell is activated by the presence of the virus, so what does it do? It runs to the lymph node. What's in the lymph node? T cells. What does HIV infect? T cells. So it's a nice strategy to find the cells that you have to infect. So the sentinels bring unwittingly HIV to the place where they want to replicate, kind of like a Trojan horse, I suppose. Another virus, an RNA virus, Venezuelan equine encephalitis virus and dengue virus, will actually enter dendritic cells and replicate them. And then, as the viruses are replicating, the DCs go to the lymph nodes, what they usually do, and then they produce and release virus, and then from there it can spread to different parts of a body. So this is actually under consideration as a vaccine strategy. If you inactivate the virus so that it doesn't spread, the DC will bring it to the lymph node, where then you have a very potent antiviral immune stimulation. Infection of these cells can also have other consequences besides these two that we have shown here. Of course, if you destroy the sentinels, you're going to dampen the immune response. It can interfere with maturation. You can even interfere with the maturation of T cells, because the dendritic cells have to produce cytokines that the T cells need in order to mature. So those viruses that do infect these sentinels are doing, for them, it's a good thing. Another component of the innate response is complement, called the complement cascade, identified in 1890. as something in serum that was sensitive to heat that would lyse bacteria when antibodies against the bacteria are present. So they said this complemented the activity of antibodies, so they call it complement. Now, complement is a major defense system that bridges innate and adaptive defenses in a different way as dendritic cells do. And it is a collection of serum and membrane proteins, many, many different components, not just a single protein, as you will see in a moment. And it can recognize pathogens as foreign by pattern recognition and help bring in antibodies to engage. So the antibodies, of course, are the product of the adaptive response. Complement is an early innate system. And once antibodies are made, complement, which is already produced early on in innate responses, will combine with these antibodies to eliminate invaders. So this can work both early and late in infection. There are actually some antibodies that are present already in all of us against various viral components. So the activation of complement early on can have benefits. We don't have to wait for the production of virus-specific antibody. So there are 4 functions of complement, 4 ways that it works. First, it breaks cells, it kills them, it pokes holes in them. These are typically infected cells that have some viral proteins on the surface that could be bound by antibodies to those proteins. So let's say an envelope virus is putting its glycoproteins in the plasma membrane, and if you've already antibodies against that, those antibodies will bind the protein, and then complement comes along and sees this antibody sitting on the cell, and it will lyse the cell. Cytolysis can also activate inflammation. It can induce the synthesis of cytokines. We'll talk about what inflammation is in a moment and why it's good for you. It can help coat virus particles to facilitate them being taken up and destroyed by macrophages or phagocytic cells. This is called opsonization. And it can solubilize immune complexes. So when you do have an adaptive immune response, you're making a lot of antibodies against foreign epitopes. When those antibodies bind to foreign proteins, they tend to clog very small capillaries. These are called immune complexes. So particularly in your kidney and brain, very tiny capillaries which are easily clogged by immune complexes. So you can get glomerular nephritis, for example, and inflammation of the kidney as a consequence. So complement plays a role in keeping these apart, not letting them aggregate. So here's one example of how complement works. You can see there are many proteins involved. These are all components of the complement cascade. You don't have to know any of them. All you need to know is what complement does. So here, for example, is an example of one of the complement components, C1q, forming a structure called C1, and this can actually coat virus particles and have them be taken up by macrophages. C1 can also recognize antibodies bound on the cell surface. So this brown line is the cell surface. So here's a viral antigen being produced. It's a transmembrane protein of some kind. Antibodies can recognize it, and then this C1 structure will recognize that, and then it activates this whole cascade that follows here with all these jiggly red arrows. So these are a variety of proteins that are made. There's a lot of processing going on here. These various soluble components here, C3a, C5, C5a, these can induce the production of cytokines by different cell types. So that's why they are called mediators of inflammation. And then finally, at the end of all this, you have the formation of a pore, the membrane attack complex. This pokes a hole in this cell and lyses it. So this is all stemming from the initial recognition by C1 of antibody bound to the surface of the cell. If it were not virus infected, the antibody wouldn't be bound here. So that's why it's specific for virus infected cells. So that's what we've talked about so far, the innate defense system, cytokines, the pattern recognition molecules, TLRs and RLRs. We haven't talked about NK cells, but they're also an important component, and the complement system. And again, they take care of most infections very early on. Your innate system is often kicking into play, and it takes care of things without you knowing it. But sometimes these are breached by viruses, and then in that case, the innate system has signaled the adaptive system that it needs help, and the adaptive system can kick in. Now, some of the main products of the innate response are cytokines, and we divide them into three primary classes. We have the pro-inflammatory cytokines, such as IL-1 and TNF. These promote the activation of various white blood cells, for example, the T cells in the vicinity of the dendritic cell. Then we have other cytokines that are anti-inflammatory. Their purpose is to balance the activity of the pro-inflammatory cytokines. So once an infection has been taken care of, these return the system to its normal basal state. And then there are a variety of chemokines whose job it is to recruit immune cells. So if there's an area that's infected, chemokines are produced, and they recruit additional cells to the area to help clear the infection. So cytokines initially are produced locally at a mucosal surface where there are virus infections going on. Those cells release material that activate, say, dendritic cells to produce cytokines and chemokines. The chemokines then pull in other cells to the area, macrophages and lymphocytes. So the lymphocytes, say, that are maturing in the lymph node are pulled here by chemokines. Initially, cytokines function locally at your mucosal surface, but if infection is proceeding, it's not controlled. You get more and more cytokines produced. Eventually, they get into your circulation, and they have global effects. So here we have IL-1, TNF-alpha, IL-6 entering the circulation, and they give you the typical non-specific symptoms of a viral infection: sleepiness, lethargy, muscle pain, no appetite, nausea, also symptoms of a virology course as well. And that's why you feel this way when you have a virus infection, because you're making these wonderful cytokines. They're circulating, and they are doing this to you. The cytokines also go into your bone marrow and stimulate hematopoiesis to make more cells to fight infections, phagocytic cells, and other sorts of cells as well. So cytokines have an important role starting very early in infection. The cytokines also induce inflammation, what we call inflammation. All right? And in particular, TNF-alpha, tumor necrosis factor alpha, is a major inducer of inflammation. What is inflammation? Redness, pain, heat, and swelling. Rubor, dolor, calor, tumor. The Romans first described inflammation way back, the ancient Romans, not today's Romans, but the ancient ones long ago recognized inflammation, that they gave it those four words which last today: redness, pain, heat, and swelling. These are all the cause of cytokines produced early in the innate response. You get increased blood flow, increased capillary permeability, influx of phagocytic cells, and tissue damage. All of that is responsible for these four signs of inflammation. And again, caused by cytokines produced by the innate response, including TNF-alpha. So here's a very nice picture that tries to integrate what I have told you so far, and in particular integrating the circulatory system, the bloodstream, with the lymph system. And of course, you know that in addition to a blood system, extensive system of blood vessels and capillaries, we also have a lymph system consisting of lymph vessels of different sizes, from capillaries all the way up to larger ones, interspersed with lymph nodes, and these are spread throughout the body, because remember, the dendritic cells go to the lymph nodes at some point to signal them that something is wrong. So in this case, we have an infection right here near the lymph vessel. It's in red color. It's labeled antigen. So imagine there's some cells here that are infected, and the virus is killing cells, and proteins and RNAs are being released. Dendritic cells, which are patrolling this area all the time, they will pick up those components, become matured. They go to lymph nodes, and there they activate the production of lymphocytes. And we have our T cells and B cells coming back to the area. How do they know where to go? Because there are chemokines produced locally by the dendritic cells. So they come back here and they start to try and eliminate the infection. In addition, we activate complement by the mechanisms we talked about. The complement activates phagocytic cells and a variety of other cells that produce cytokines and chemokines as well. And a lot of those, here's your IL-1, TNF-alpha, interferon gamma, they have effects on the blood vessels. So they make the blood vessels more permeable. They cause migration of macrophages from within the circulation to the infected area. So that's how we have this orchestration between the lymph and the blood system in an infected area. And again, the consequences of all these cytokine releases early on is inflammation, the swelling, redness, heat, and pain. Inflammation is good for you. It stimulates potent immune defenses. Cytopathic viruses, that is, viruses that kill cells, they cause inflammation because they damage the cell. So if a virus kills a cell, you have bits of dead cell floating around and virus parts, and those are picked up by the dendritic cells, and that's good. So the dendritic cell can sense that there's an infection. If the cell isn't being killed, it's very hard to sense that it's infected. So that's why inflammation is good. It means that the dendritic cells have sensed something foreign, they're producing cytokines and so forth, and inflammation. So inflammation, cell and tissue damage activates the innate response, leading to inflammation. So viral genomes from cytopathic viruses typically encode proteins that modulate the induction of inflammation, as you will see from Dr. Silverstein. They have to, otherwise they would be eliminated by the inflammatory response. Adenoviruses, herpes viruses, pox viruses, the viruses with larger DNA genomes, where they have a lot of room to spare, they encode extra proteins that specifically antagonize innate responses. They don't want to be sensed. They want to replicate. Some viruses don't activate the innate response, the adaptive response very well, because they don't stimulate the inflammatory response. Remember, you have to engage the innate response, you get inflammation, and then you get engagement of the adaptive response. And some viruses don't do that because they're not good at stimulating innate responses. And these are usually viruses that don't kill cells, the non-cytopathic viruses. And as I said, if you don't kill a cell, you don't release material that can be picked up by the dendritic cells. So cells are not damaged. They don't die of infection. There's no cytopathic effect. So you don't have an intrinsic response, and you don't have a very good innate immune response. So if you don't have an innate immune response, you don't have those cytokines produced, you don't have inflammation, and you never get to a good adaptive response. So the interaction of non-cytopathic viruses with the immune system is very different from cytopathic viruses because of all this. And these are typically arenaviruses like Lassa fever virus. They don't kill cells. Paramyxoviruses like measles viruses. They typically cause persistent infections because they can't be cleared. You don't have a good innate response, and therefore you cannot clear the infection. Clearing an infection requires a really good adaptive response. So again, why don't you make a good innate response? Because you don't make material that can be sensed by the dendritic cells. So the lesson, the inflammatory response, heat, swelling, redness, pain, reflects the communication of the innate and adaptive immune defenses. If you have no inflammatory response, you're going to have a bad adaptive response, and that's because the cytokines aren't produced that link the two processes together. This is one reason why we use adjuvants in many vaccines that don't replicate. So there, as we will see later in when we talk about vaccines, there are many vaccines that are inactivated, so they don't infect you, and so they don't kill cells, right? Because they can't get in the cell and replicate. So they're not sensed very well by the innate response, and you don't get good inflammation. And if you don't get good inflammation, you don't get a good adaptive response. And a vaccine's whole modus operandus is to make a really good adaptive response. So sometimes we add adjuvants to these vaccines, and the purpose of those adjuvants is to stimulate inflammation, because they have materials in them that are sensed by the innate system and induce these cytokines that you need to make a good adaptive response. Only a few vaccines in the US have adjuvants so far. In Europe, they use many more. And there's a lot of opposition to adjuvants, but it's probably not well justified. We'll talk more about that in vaccines. So here's a nice summary so far of what we're talking about. We're about to move into adaptive responses. But let's go back and remember we have a nice mucosal surface, epithelial cells infected by a virus. The virus-infected cells release, if the cells are killed, they release products which are picked up by immature dendritic cells. The infected cells can also produce cytokines that help the DCs mature, because remember, the DCs have receptors for those cytokines. The DCs mature, they go to the lymph node, and they present viral peptides on their surface to the T cells in the lymph node. The T cells mature, and then those go out into the circulation, as well as antibodies produced by B cells. So here is our one-slide summary of the adaptive response, which probably Moshowitz spent 2 weeks on or more. We have two arms that we have to keep in mind. You introduce some foreign antigen, protein, viruses, bacteria, parasites, fungi, into a vertebrate. You have a humoral response and a cell-mediated response. The humoral response, of course, is the production of antibodies by activated B cells. These antibodies can combine directly with virus particles and render them non-infectious. So the antibody is important for preventing infection. But if you are already infected, an antibody has less of an importance. It's good if you already have the antibodies and you're infected. It will prevent you from being infected. But when you are infected for the first time by a virus, it takes 2 weeks or more to make antibodies, and so they don't really have a lot of role in your recovery. What is important for recovering from a virus infection is the cell-mediated response shown on the right. And that consists of course, helper cells or cytotoxic T lymphocytes. So these proceed from immature cells in the bone marrow, they go to the thymus, and then finally the lymph node. And of course there they can be activated by dendritic cells, which say, look, I have some antigen, and if it's a foreign antigen, these cells were mature. So the T helper cells, the CD4 cells, produce cytokines. That's their main role. They produce cytokines depending on the subtype that either help B cells mature to make antibodies, or they make cytokines that help CD8 cells mature into cytotoxic T cells. So the CD4 TH cells have a maturing role. The CTLs, of course, kill infected cells. They recognize when a cell is infected and they kill it. The idea being we're going to stop any more virus replication. Of course, that damages cells, and it is a component of the pathology of infection, but it does limit infection. And in fact, CTLs are crucial for recovery for most virus infections. So if you want to eradicate infected cells and virus particles, you need an adaptive response to clear the infection. An innate response cannot clear an infection once it gets beyond a certain stage. The effectors are antibodies, as we've said, that bind viruses and decrease the number of infected cells. And if you remember, they can also work with complements. So if you have an antibody against a viral protein that's on the cell surface, complement will recognize that and lyse the infected cell. The T cells will recognize infected cells. And of course, they respond to viral antigens on the dendritic cells in the lymph nodes. That's how they get activated in the first place. They kill virus-infected cells. And as I said, the helper T cells make cytokines to either aid in antibody production or the maturation of CTLs. So distinguishing the infected self from an uninfected self is done by the innate immune system by the pattern recognition receptors, the toll-like receptors, the Rig-I-like receptors, even complement by recognizing, for example, antibodies bound to the surfaces of cells. We haven't talked about NK cells, but they have their own way of recognizing self and non-self. The adaptive immune system recognizes this difference by antigen presentation, which we're not going to discuss at all, and you won't be responsible for. But Dr. Silverstein will talk about ways that viruses antagonize antigen presentation as a means of evading adaptive responses. So you can find out more of about that in Chapter 4. Let's talk a little bit about antibodies that are made by the adaptive response. As I said, these are essential in the defense against many infections. When you get vaccinated, the reason that you are protected is because you make antibodies against the particular virus. These do what we call neutralize virus particles in the blood. So if the virus is spreading in your blood, you make antibodies. The antibodies will combine with the virus, and that will block them from infecting new cells. There are also antibodies made at mucosal surfaces, the so-called secretory antibodies. So if you have a respiratory virus infection, these can bind the virus before it can even infect your mucosal epithelium. That's what I said right there. And some, even though I told you that in general neutralizing antibodies protect you, there are some examples where they're important for recovery from infection. So nothing is ever absolute in science. Nothing is ever black and white. There are always exceptions. So if I say to you, antibodies are typically needed for protection, that's true, but there are always exceptions. So there's some examples where antibodies are needed for recovery. So here's an experiment which shows that antibody protects against polio infection. This is a passive antibody. So remember we talked about passive viremia a while ago. Passive viremia is when you inject virus into an animal. The virus that first appears in the blood, say, is passive viremia because it's not resulting from replication. So these are antibodies that you inject. They're not produced by the animal. They're just simply antibodies you've injected. So in this experiment, animals have been inoculated with increasing amounts of antibodies. So the bigger number, more antibody. Here's a control animal with no antibody. And then they're infected with poliovirus. And this is percent paralyzed. So almost all the animals are paralyzed when you inject them and there's no antibodies given. And then as you increase the amount of antibody, you protect them from paralysis. Very clear experiment showing that you can prevent infection with antibodies. And this is the basis of vaccination, of course. So how do antibodies work to prevent infection? So here we're illustrating the answer to this using a picornavirus, rhinovirus. And we're showing how this virus gets into cells. It binds receptors on the cell surface. It's taken up into the cell by receptor-mediated endocytosis. And the RNA is uncoated as the pH in the endosome drops. And on the right are a series of scenarios showing how antibodies that bind rhinovirus could block infection. So remember that you're infected with rhinovirus. Your adaptive system produces antibodies that recognize these virus particles, and they will bind. There'll be a whole set of antibodies produced to all the proteins, and some of them will bind to the surface of the virus itself, the virion. Some of these can block attachment of the viruses to cells. If you have enough binding in the right places, you will block attachment of the virus and thereby block its infectivity. So that is blocking before infection begins. You can also block later on. You can have the antibodies binding to the virus. The virus is still taken up into an endosome, but uncoating is blocked. So there are examples of antibodies that do this. So the particle gets all the way into the cell, but then when the pH drops, the antibodies prevent the changes in the particle that are needed to get the RNA out. And finally, you have antibodies that aggregate viruses so that they're so large, these aggregates are so big they cannot be taken up into the cell. So 3 different mechanisms of neutralization. This is how antibodies block infectivity in various parts of you, either your blood or at mucosal surfaces. So what are these antibodies recognizing? When antibodies are made, of course, they're made against viruses that are broken up by antigen-presenting cells. The peptides are presented to B cells, so you make antibodies against all the possible peptides of a virus. But the ones that are important for us are the antibodies that bind to the surface and can block infection. And these are 2 examples of the epitopes recognized by these antibodies. So an epitope is a short amino acid sequence that is recognized by an antibody, and all viral proteins have epitopes in them. But the ones that we care about in this discussion are the neutralizing epitopes. That is the ones where an antibody will bind and block infection. So that's what we're showing here. Here on the left is a structure of rhinovirus, and we're showing in the lighter colors some of the neutralizing epitopes. That is, these are short sequences to which antibodies will bind and block infection. So these are on the surface of the particle, and they're in very specific places. You can see not every sequence is a neutralizing epitope. So that's for a virus with an icosahedral capsid. Here on the right is the hemagglutinin of influenza virus. You remember, this is the molecule on the surface of the virus that binds to the receptor to get the virus into a cell. And then it also is involved in fusing of the virus in cell membrane at low pH. And the colors show you the neutralizing epitopes of the hemagglutinin. These are all mainly located at the distal tip of the hemagglutinin. So the receptor binding area is right up here. And these colored areas are all neutralizing epitopes. So antibodies would bind to these portions of the HA and thereby block either attachment or fusion in the endosome. Now, one of the reasons why influenza is a recurring problem is because these epitopes mutate every year. You have amino acid changes in these neutralizing epitopes, so the antibodies that you make one season against influenza virus that neutralize that virus very well, the next year will not neutralize it. Even just 1 or 2 amino acid changes in one of these epitopes will make the antibodies ineffective. And that's why, with time, we have to be immunized against the new strains of influenza that arise. All right, so those are neutralizing antibodies in the epitopes that they recognize. Now, cell-mediated immunity, as I've said, is essential for clearing most virus infections, and this is what we call the Th1 response. It is generated by a synapse formed between the cytotoxic T lymphocytes and the target cells that are infected with viruses. And the CTLs bind the target cell. They recognize foreign antigen on the surface of the targets of the infected cell, and they lyse them. And there are 2 ways that the CTLs lyse them. One, they transfer granules to the target cell that have these proteins, perforin and granzymes, and they poke holes in the target cell, effectively lysing it. And also, they can induce apoptosis. So the engagement of the CTL with an infected target cell can turn on apoptotic pathways, which will also cause the target cell to die. So here's an experiment where we look at the role of antibodies versus cell-mediated immunity in protecting monkeys against monkeypox infection. So what we have done here is we have immunized these animals at day 0. And then we have done something to their immune system here, either nothing, the control, or we deplete the B cells, or the CD8 cells, which are the CTLs, the cytotoxic T cells. So the B cells, of course, making antibodies. The CTLs is the cellular response. So we're going to tell which one is important. So in these animals vaccinated day 0 and no manipulation, they make a nice antibody response on day 22. If you take away the B cells, the antibody response is dampened. So this is antibody titer. It's neutralizing titer, so the larger number is better. And the ones depleted of CD8s have slightly reduced, but not a bad antibody response. Then we infect these animals with monkeypox on day 28, and we look at fatal infection and high titer replication. So here, these animals who were immunized and not manipulated are all protected from lethal infection. If we take away the B cells, 3 out of the 4 animals die. If we take away the T cells, the animals are fine. So here you can see if you have antibodies ahead of time, you're protected from infection. It doesn't matter if you have T cells, the antibodies are really what's important. But sometimes both antibody and cell responses can play a role. So here's another experiment to show you that. We're looking at virus replication on the y-axis, either by RNA or plaque-forming units here. And we have animals who are infected. They're all infected at day 0. And we have a control group here in the closed circles. And here's the titer of virus in the serum. It goes up, and then the titers go down, and about 30% of the animals die. If you deplete antibodies from these animals, they all die, and the virus cannot be controlled. So in the normally unmanipulated animals, they eventually control, the ones who survive control virus infection. But if you take away antibodies, they cannot clear the infection. So that shows that antibody is also important in this case. If you deplete CD8 cells, you have 85% death, a little more death than the controls. But these guys are pretty good at controlling the levels of virus, presumably because they have antibodies. So that's in the serum. In the spleen, you can see the control group has a similar pattern of virus production. But if you take away either antibodies or CD8 cells, these the virus in the spleen cannot be controlled. So the control of virus replication differs according to where you look, blood versus spleen. Same thing with the brain. If you take away antibodies or CD8 cells, you have a hard time controlling virus titers. So as I said, even though we say that CD8s are important for recovery and antibodies important for infection, there are always exceptions to the rules. So for some infections, the CTL response is more important than antibodies and vice versa. That's what I've tried to illustrate by those couple of experiments. How do we make sure that we have the right combination? So antibodies, cellular responses, how do we modulate that? That begins in the lymph node when the mature dendritic cells come in and talk to the T cells and other lymphocytes that are present there. And it all has to do with Th cells, the CD4 positive lymphocytes, which remember are just making cytokines. That's their role. They can make cytokines that stimulate the production of B cells or CTLs. And the direction really depends on the virus. So every virus and host combination has a slightly different need for B cell versus CTL, and that's controlled by these Th cells. Remember, these cells in the lymph nodes make contact with dendritic cells and also macrophages that are patrolling and bringing in foreign antigen. They exchange information. This we've talked about already today, peptides and cytokines, and that causes the differentiation of the CD4s into either Th1 or Th2 cells. Th1 cells are those that secrete cytokines that make the T cells differentiate into cytotoxic T cells. And the Th2 cells make cytokines that make B cells make antibodies. So you see there's a very early decision point where you have to decide you're going to go the B cell route or the CTL route. And that depends on whether you make Th1 or Th2 cells. And there seems to be a yin-yang relationship. You have a lot of Th1 cells, low Th2, and vice versa. You don't make both at high levels. So how do you decide whether to make Th1 or Th2 cells? Well, most viral infections, as I've said, are cleared by the Th1 cell, the cytotoxic T lymphocyte response. Th2 cells activate the antibody response. This is not a good response for most virus infections. Remember, with a few exceptions that I showed you, most virus infections to be cleared need a CTL response. This is good for bacterial and worm infections to make antibodies, but not for virus infections. So what is the response do you get? What mixture of both gets turned on and how? And it's the mixture of cytokines that are produced by infected cells or sentinel cells. So you have a precursor cell that can either go to Th1 or Th2. It's an immature CD4 positive Th cell. And remember, in the lymph node, these cells are encountering the dendritic cells and being activated by them. And the mix of cytokines that are produced determines whether they go to Th1 or the Th2 route. So if you're making, for example, IL-2, 12, and interferon gamma, it pushes you to the Th1 side. Or if you make IL-4, 5, and 10, you go the other route. You don't need to know what particular cocktail of cytokines pushes it either way. You just need to know that the decision to go Th1 or Th2 is based on which cytokines you get. Now, interestingly, look here, the suppression of the Th1 response is done by some of the cytokines that induce the Th2 response. So that's why I said this was a yin-yang relationship. You had either one or the other high, but not both. And again, the kinds of cytokines produced are presumably detected by the dendritic cells according to which pathogen is present. So they can sense that, and then the CD4 T cells respond by making the right cocktail of cytokines. These adaptive responses also provide memory. This is very important. So that if you're infected later on, you get a more rapid and specific response. So the adaptive response takes 2 to 3 weeks to engage fully. If you're reinfected, it's much faster because you have memory. Innate responses don't have that. They're always on. They respond quickly. They don't need to have memory. And this, of course, is the basis for vaccination. You're immunized, and then you encounter the pathogen some other time, and you respond quicker, so you prevent infection. So you make antibodies very quickly. You block infection before it starts. And that's why antibodies will prevent infection, whereas if you have a first encounter with a pathogen, you need the CTL response to clear it. So here's an experiment where we look at antibody production after time. So we inject an animal with antigen A. We have a primary response 3-4 weeks after injection. It's kind of modest. And then the antibody levels return to low levels. If you get infected, or in this case we're injecting antigen A 7 weeks down the road, you have a very specific and much more effective antibody response against A. So the curve goes up higher and much more quickly. And eventually that goes down as well. So that's an example of memory. You have memory cells that will respond to this antigen very quickly and more robustly. There is also cellular memory as well, not just antibody memory. So there are B-cell memory and T-cell memory. That's shown here. Here we have infected an animal with a virus. The titer rises. This is virus in the spleen, and then it goes back down again. At the same time, that animal is making cytotoxic T lymphocytes. Those are the green bars, and they are specific for the virus. So if you take the CTLs out of the animal at these different time points, you can show that they will lyse virus-infected cells. But eventually the activity of those go down as well. But still circulating in the blood are epitope-specific lymphocytes. So they don't kill cells, but they will recognize viral epitopes. So that means that there is memory towards those CD8 lymphocyte epitopes. So again, there is not just memory to antibody-producing cells, but also CTLs as well. And we'll talk more about memory when we consider vaccines.